From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on July 11th, 2024. Hello, Micronauts, and welcome from the campus of the California Institute of Technology in Pasadena, California. In a moment, I'm going to be fully introducing my guest, Dr. Margaret McFall-Ngai. But for now, I'd just like to bring you in the fact that I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 49th episode of Matters Microbial. We're almost to 50, if you can believe it. Those of you who watch and or listen and spread the good microbial word to others, many, many thanks. 50. As this episode is being recorded, as I mentioned, I'm at Caltech in Pasadena, California, a very impressive place. My own philosophy about our microbial world is one of microbial centricity, as you can see from this button. In addition, I believe it's difficult to think of any organism without its microbial partners, which inspired me to commission this view of a human face made of microbes. Thus, I am a microbe biologist. Sorry about that. The Latin translates to small, powerful, and numerous, they rule the world. And finally, from one of my introductory writing courses on symbiosis, we are indeed all walking ecologies. All of this fine artwork was commissioned by me and created by Caitlin Reese. For many years, my views have been inspired, motivated, and fine-tuned by association with many truly first-rate biologists. My interest in the Squid-vibrio symbiosis introduced me to Dr. Ned Ruby and my guest today, Dr. Margaret McFall-Ngai. And that tractable system that can be manipulated on both sides quickly led to a fascinating view of every living thing swimming in a sea of microbes. They're part of us, evolved along with us, and modify us just as we can modify them. So it's an honor to introduce Dr. Margaret McFall-Ngai of Caltech to the Quality Quorum for today. I cannot think of a better person to talk with us about the philosophical implications of the microbial universe in which we live. It's always a pleasure to see you, Margaret, and always an honor. Welcome to our Quality Quorum.
Thanks for the opportunity, Mark. This is really going to be fun. I really appreciate it.
Yeah, it's a little bit of a task getting this to work, hence this particular design, but I'm glad to be sitting next to you and actually visiting with you.
Yeah. Well, and I want to make one, I'm not a microbiologist, and I often tell people, when I'm invited to give talks in microbiology departments, people have to understand that I'm a huge microbiology fan, as you'll learn, but I was actually trained as a comparative animal physiologist biochemist.
That's indeed right. If you look on the Wikipedia page, they call you a biochemist, which is pretty amazing. But Margaret has had a very interesting holistic view about things. It's been very influential to my own thoughts about our association with the microbial world, and more particularly the way that I teach this topic, because I think there are some things missing that we can address that'll help it. It's a much richer, wider world than we expect, and that's primarily due to the microbes that are with us. By the way, a wonderful quote, I don't know if you've heard it, Margaret, from Dean Martin.
Yeah, no.
Dean Martin, the old singer, was a friend of Frank Sinatra's. And at one point, he's reputed to have said, "It's Frank's world. We just live in it." And I would argue it's a microbial world, and we just live in it.
Yeah, that's wonderful. That's really wonderful.
So I think a good place to start is you mentioned your background is essentially a comparative animal physiologist and biochemist at UCLA.
That's right.
Now, I was a baby overlapping with you there, but I never ran into you because I wasn't taking those courses.
Right. Yeah, I was there, and actually I was studying already symbiosis with bacteria as a graduate student. And my study site was the Philippine Islands, and I worked on a small fish that had luminous bacteria associated with it.
The Japanese lanternfish.
No, not that one.
Not Monocentris japonicum?
No, this was something called lignathids. The lignathids are, they're actually probably the major and most abundant fish in Southeast Asia. And they occur in very large schools. And if you have that Japanese fish cake that's white with the pink around the edge, that's usually a derivative of a process with lignathids. That's fish cakes.
And I was influenced by Len Muscantine, and I know you had some associations with him as well.
Yeah. Actually, so Len was a very distinguished faculty member across the hall from the lab that I was working in. And he was studying symbiosis in corals, in corals and their zooxanthellae symbionts. And, Len was just an amazing person. And he and I became very good friends. And we went back to MBL and taught together as part of the Boston University Marine Program 3 times. And it was really fun. He was a tremendous person. And I would say probably during his career, the most famous coral reef biologist, and really contributed a huge amount to the field.
Absolutely. I mean, I'm thinking about my own experiences there and at UCLA, and we had a good time. I recently had a situation you're aware of. I had this Mohs surgery to remove a lesion, and they got to show me the histology, and Jim Lake came rushing back into my brain from my own histology class. And I remembered, which is good to know.
Yes. Yeah, UCLA. I actually had an opportunity to go there yesterday because I'm heading off to Berlin to the Wissenschaftskolleg, which is a think tank. And my goal is to create a manuscript that I hope to get into a high quality, very conspicuous journal that calls for remodeling biology education to integrate macro and microbiology, because I think it's really essential. You can't graduate anymore with an undergraduate degree in biology with a single lecture on the microbial world. It just doesn't work. So I think we have to move in that direction. But I was over at UCLA they needed my transcripts for my German visa. And so I had to go over there and pick them up. And it's a beautiful place. Memories, flooded in. It was really fun. It was really fun.
I have a lot of shame about my youth personally, but I'm haunted by the memories. But it was a wonderful opportunity. It's true. And what I really want viewers and listeners to think about, there are many, many paths to the microbial and macrobial sciences. And it's important to find the things that you feel passion toward and the things that really interest you. And too many times I hear people talking themselves into and out of things. And that's nothing that Margaret has ever done. She's always done what Margaret wanted to do.
Yeah.
And it's paid off.
Well, I think we are so lucky in this generation and the following generations, because we actually are in a major revolution in biology. And we get to be on the cutting edge, at the frontier of this phenomenal area. And, the way I look at it is that over the last 3 centuries, there have been 3 revolutions in biology. One is, Darwin's theory of natural selection, and that was in the mid to late 19th century. Then in the 20th century, in the mid 20th century, there was DNA discovered as the genetic material. And then in the 21st century, we have found through changes in technology and whatnot, we've been enabled to discover a world we couldn't have known. And that's the microbial world. And hands down, used to be thought, for instance, that there were 7,000 species of bacteria. And that's the same number of species as there are species of snake. And so there's an absurdity to it. And so, with Terra Oceans and the Human Microbiome Project and other such things, we now know that by far and away the microbes are the most diverse organisms on Earth. And they they are at the basis of health of absolutely everything. And so we really do have to respond to that. And we as biologists have to begin to integrate macro and microbiology. It's downright dangerous that with what we're facing environmentally, if the the macrobiologists don't recognize microbiology, and the microbiologists don't recognize macrobiology. One of the problems we have, and one of the problems we have to solve, is that beginning in the 1950s with the discovery of DNA, biology went into deep silos. They went into deep silos. And so there are microbiologists, there are cell and developmental biologists, there are ecologists, there are physiologists. And there's a lack of communication among those different groups because they were trained in very narrow ways. And so that's one of the other things I'm going to be calling for. And the analogy that I often make is that in chemistry and physics are great and growing fields and lots going on in those areas. But when they teach introductory chemistry and physics, that chemistry and physics, they have defined the major principles that they want all physicists to know or all chemists to know and understand. And so one of the exercises I've done is to go back to the table of contents in physics and chemistry books from when I was a kid. And the big table of contents is the same today as it was when I was a kid. And that just reflects the fact that they found these foundational principles that they want all of their physics students to know. And then they can go off and go into quantum physics or plasma, they can go off into all kinds of other branches of physics. But, what we've got to do is we've got to teach a broader course. We've got to do what chemists and physicists have done. If you look at a textbook of biology from when I was a kid, it's completely different. And what we do is we're bricolage. We just, every time we find something new, we poke it into the middle of an existing tome. And we really, really need to think about basic principles, like how do organisms get energy? How do they seemingly defy the laws of thermodynamics? What are the limits on cell size? I mean, there are things that are basic without all this memorization of trivia. We need to do that in a big way.
I call it like a 3x5, just telling you, a 3x5 card stack in your head. Mm-hmm. And that's how a lot of students think about things, and they don't retain that information. Mm-hmm. Just like when I took chemistry, I would do what I could for first semester, but I didn't remember things for the next semester. And in biology, you can't afford to do that because it's all interconnected. And that's one of the things that I'm happy about, and I hope you enjoy this story. Please forgive me. I had some students a few years ago who took microbiology from me in the fall, and then they took animal physiology in the spring. And as part of their project, they had an independent project, and they were working with earthworms, and they're kind of like brainstorming, and they came to tell me this story, that they said, well, what about humidity? What about temperature? And then one of them said, we haven't even considered the microbial components. And they both stopped, looked at each other and said, we drank the Kool-Aid. And they did, because it is part of the whole world. Mm-hmm. And I think that's a good way to think of it. We talk a lot about ecology and working together and, Mother Gaia, if you go with Lovelock stuff. But the most important thing to remember is we're all connected, and there are echoes evolutionarily of all this. I often say that the hand of Darwin is on all that lives. Mm-hmm. And though I find when I talk to people who are trained as, say, and I hate this expression, by the way, big bio and little bio. I've always hated it because it's all biology. And I don't know if you know this, but when I took biology at UCLA, you could take it for kind of a cell and molecular, or you could take it for the other specialty. And because I'm a stubborn so-and-so, I took the population and organismal biology courses for people in that specialty. Martin Cody kicked my behind in ecology.
I took that class.
Yeah.
Bio 122.
Yes.
Flute music across the board for the entire time. In other words, ecology is incredibly mathematical. So
Henry Hespenheit, who really made me appreciate evolution.
Yes.
And I would have missed those things if I would have stuck with, quote, the CMB major, and I have always maintained, and I think students have a right to choose whatever major they want, don't get me wrong, but I think about what you're losing.
Yeah. Well, yeah, and evolution is the glue that joins all of biology. And so that's the thing. If you understood, the other thing is that oftentimes biologists don't understand the history of the biosphere. How can you not understand the history of the biosphere? It's really critical. And so what the goal would be is to create a foundation for biology, a framework that people could then, once they go off into specialization, they can hang those things on that scaffolding. And it's such an interesting thing, because I think biology, the other dimension of that is that, when I took chemistry and physics, I took chemistry and physics with a chemistry and physics majors. It was just the way it was done when I was in school. Chemistry and physics majors didn't have to take biology. But we know that many, if not most, of the challenges of humankind have biology at their base, but chemistry, physics, engineering, and math at their solutions. And those people don't know biology. So that would be another goal, is to incorporate, is to bring those groups of people. Now, there are 2 universities for sure that I know that require biology of all of their physics majors and computer science majors and whatnot, and that's MIT and Caltech. Yeah, and there might be others. It's just that I know that those 2 do.
You can also get a certificate in piracy at MIT if you take their PE courses that are relevant. I'm not making that up. I'll put a link in the show notes.
Oh, that's wonderful.
I know, I want one. But this is exactly right. I've often said that my goal, and I'll get this a lot, I'll hear criticisms of poor Doc Martin that I'm not going, quote, deep enough into microbiology when I have 1 course. And I could make the same argument for introductory biology of anything, couldn't I? Mm-hmm. And so what I try and do is try and create, as you say, I call it like a holiday tree, and then the factoids are the ornaments that you hang about it. But more importantly are the connections between different parts of those trees. Mm-hmm. And we tend to forget those things. I mean, as you say, we live in remarkable times. The tools that we have are amazing. Mm-hmm. And sometimes students, I don't think, appreciate that. When it used to be so difficult to sequence a fragment of DNA, and now it's something that costs like a few $dozen, depending on the size. So the point is, we can really ask and answer fascinating questions now. And we have to stay away from those 3x5 cards and really think about, I hate to use the term critical thinking because that gets misused a lot, but really being analytical about what you see. You've met Ken Nielsen now at UC.
Oh, he was on my PhD committee. Yeah.
So you remember that Ken had, and this is where I got the term, a Wunderkammer of little items he had that he would hand you and say, explain that to me. And that always struck me. Wow. So I do the same thing with stromatolites. Mm-hmm. I have little pieces of stromatolites. The people that believe in crystal power also like stromatolites, so I can get them for cheap.
Oh.
and I'll hand them out to students, and I'll say, tell me what this is. And they'll say, well, it's got some bands on it, right? I said, well, tell me how that happened. Go read stromatolites and come back. You're holding a piece of history in your hands. How old? 500 million years?
Yeah, isn't that amazing? That's really amazing. It's such a fun time, because I think we have this opportunity, and it's sometimes hard because there are people who are really threatened by it. And so, I once had the privilege of being down at UC San Diego, and I was in a committee with the brass of UC San Diego, and I mentioned that, for my money, integration of macro and microbiology is the next frontier, or one that should be evolving. And there was a guy, this was like 10 years ago, and there was a guy from Salk who was a very, very, very famous neurobiologist who said, the microbes have nothing to do with the brain. And I said, you know what, I'm on sabbatical at Caltech, and Sarkis Mazbanian and I just taught a course in which we went over the first publication showing that the microbiome shapes the mouse brain. And I said, so you might keep an open mind. But there's this siloing, that people are comfortable in their silos, and it's very hard for them to be open to other things. But, I think that's where we need to go. And I'm really obviously very passionate about this. And part of that comes from the fact that I had this great privilege of teaching a course when I was on sabbatical here with Diane Newman. And it was I had taught intro bio 18 times before that at UCLA, at USC, at Wisconsin, at Hawaii. Because I love teaching intro bio. And this was an opportunity to partner with a microbiologist. And she would get up at the beginning of every course and say, okay, what is behavior in microbes? And she'd say, okay, there's on this microorganism, there are these things called MCPs, which are methyl chemotaxis proteins. And what they allow the microbe to do is sense things in the environment to which they will maybe chemotax or not chemotax. And so what happens is those molecules in a portion of the cell are sensory. And then there's an integration across the cell, usually phosphorylation cascades and the like, that says, okay, in my history, is this a good thing or a bad thing? Should I move toward that or move away from that? And then at the other end, there's a flagellum. And so you have a sensory system, you have an integration, and you have an effector. So Diane would go into this whole thing, and it would in some depth. And then I'd get up and I'd say, okay, you guys, just let's start off and say, what happens when you're multicellular? Well, there are three kinds of neurons. There are sensory neurons, there are interneurons, and motor neurons. There's nothing new under the sun. What the organism has to do is sense the environment, integrate the information, and respond appropriately. And so there are three kinds of neurons that do that. And so these basic principles are there. And I think that I'm hoping that there's more interaction between microbiologists and macrobiologists than there are. When I, would go to the American Society of Microbiology meetings, for which I was the incoming chair, and past chair for 9 years, I would often be, I would think, one of the only macrobiologists at the whole meeting of several thousand people, right? And that's also true when I go to a developmental biology meeting. I will be the only person who studies symbiosis. Now that's changing some. I mean, it's getting to the point where people are recognizing that this is something that has to be factored in. But how do we do this? How do we do this? How do we change universities and making, departments less siloed? How do we change funding agencies and make funding agencies less siloed? And, actually Harold Varmus suggested that there would be 3 or 4 instead of, 20, however many divisions there are or institutes there are at NIH. He suggested 4, and one of them was Microbial. And
do you know Varmus very well?
No, I don't know him at all.
So he's a hero of mine, and you need to hear this, friends. So when I was in graduate school all these years ago, when dinosaurs ruled the earth, and I happened to work on a small virus of Rhizobium melilodi that we were using as kind of a gene transfer agent. And Harold Varmus came to visit Stanford's campus, very famous scientist, and we're walking across to have lunch, and everybody is jockeying for position to talk with him, and I'm not comfortable with that sort of thing. I guess I'm a shy person, actually.
Oh, yeah.
This is acting, friends. Anyway, what's important to remember is that I was kind of like holding back, and then Varmus came over to me and he said, so what are you working on your PhD about? And I started to explain it to him, and another person whose name you know, and I won't repeat it to you, not even later at lunch, said, well, that's not interesting. Varmus lost it.
Oh, my gosh.
He made me sit next to him at lunch.
Oh, wonderful.
And he talked to me about all my stuff and the history of molecular biology, and I'll never forget that. So you follow your microbial heart in my case, your macrobial heart in yours.
Well, so here's the way I feel about it. I feel that we need, and this is heresy for most people, we need to go back to organismal biology, because biology is about organisms. It's not about chemistry. It's not about physics. It's not about molecular biology. It's not about, those are the basis of how organisms work. And then there are, then they get into populations and communities and whatnot. And so, but the organism, biology should pivot around the organism. That's the unit of selection, evolutionary selection. It just so happens that the vast, vast, vast majority of the world is microbial. It just, and, you know, it's one of those things is once you accept that, like this was for me, you know, I know some microbiology because I've been hanging out with you guys for a long time. But I mean, it was to sit in the class and have Diane get up there and say, how does transcription work in bacteria? And then I would have to get up and say, okay, now you're gonna transcribe something, and how do you make the RNA stable? Because it has to go out into the cytoplasm to the ribosomes. You put on a 5' cap and a poly A tail, you know, and so there are these nuances that happen, but you know, microbes invented almost everything. And every time we think, like, oh, they don't have introns, oh, they don't have multicellularity, oh, they don't have, you know, we're wrong. And so what we've got to do, I think, is be open to seeing how things have been modified over time.
Do you know the physics statement that I like so much? Constants aren't and variables don't. And no, that's not true or anything. But the point is, people get used to the idea of the way things are, kind of like from the movie Babe, the way things are. No, the world is so incredibly complex.
Yeah.
And microbes, I say first evolved, last extinct, because that's me, microbial centrist. But there are echoes in us of them, and we learn from those things.
Oh, yeah, we are, you know, we are, a large proportion of our genome is derived from the microbial world.
Do you know the lateral flagella issue in Vibrio parahemolyticus?
Swarming thing?
Yeah.
Yeah.
So it's very interesting that Vibrio parahemolyticus, that causes food poisoning in a lot of cases, exists with a single polar flagellum to swim through liquid, and then dozens of what are called lateral flagellas to move across the surface. And I want you to imagine all of those lateral flagella spinning, and they're kind of like judo flipping one another. And that's how they move across the surface. So Linda McCarter and Bob Belis many years ago were able to determine what it was the cell was sensing. So we're used to the idea when one cell touches a cell, and we have the integrins and we have cadherins, and we can figure all that out. But turns out, just to reference it in terms of watching our time, anything that impedes the rotation of that polar flagellum, that was the signal. And then if you put it like in a very viscous solution, they make lateral flagella and liquid. I still don't know how that changed, that dynamometer, bringing you back to physics, that dynamometer impeding rotation, how that's converted to a change in gene expression. But that's fascinating to me.
Oh, it is. It's fascinating. I mean, we have so much to learn. It's just a new day. It's a new day. And I'm kind of jealous that I'm, you know, in the last 10 years of my career, and it's a tough, you know, to think, oh my gosh, but it's going to be fun to watch as things develop. It's going to be really fun. And it you know, it's so funny because that guy about the brain, he said to me, oh, you know, when he said, this guy down at Salk who said, you know, that microbes have nothing to do with the brain, his next sentence was, and the microbiome is just a fad. And I looked at him and I said, you know, it's not a fad. It's the way the world works. I mean, we've got to accept that. We really do. It is. You know, and like I said, I'm an animal biologist. And, you know, it is the way the world works. And to properly understand the biosphere, it requires that we do this. integration.
And you know, at many institutions, and well, that's not true. I am making a judgment, but I mean nothing negative by it. What I am saying, at many institutions, they silo into what they call, I cringe at this, big bio, little bio, the idea of environmental and organismal and sometimes ecological versus cell and molecular, when it's all biology. And we're enriched from this. Historically, we always see more from getting other points of view in.
Yeah, and you know, I really, that's really true. And I will forever be grateful to being a postdoc with George Slammerell. And the reason why was because his whole life was studying at the biochemical and molecular levels how organisms live in the ecological niches in which they live. And so he spanned the hierarchy of life by you know, and that I think is just an amazing thing, because we need to understand how climate change is going to affect everything. And I have the privilege of being part of a group that is going in Europe. They're going to have a big meeting at the MBL on the cell biology of climate change. And you know, this is something that, you know, the cell and molecular biologists haven't been thinking as much about as they might. And you know, it's going to be very exciting.
You mentioned the idea that there's a relationship between studying microbes and some of the impending climate crisis. Most people, when they think about microbes, think about them reproducing so quickly and being so adaptable and so protean in form. What kind of effects could climate change have on our microbial communities, and could that be a bad thing?
Well, that's a great question, Mark. I mean, one of the things is that not only is climate change going to influence the microbes, but the microbes really are going to change climate change because they are foundational. And the best example that I can think of right off the top of my head is the permafrost. And so as the permafrost melts, methane is going to be released into the environment, and that methane is produced by the bacteria that live in those habitats. As far as the microbes are concerned that are living in and on us and associated, that they might be, because they do divide so quickly and whatnot, and it's in their best interest to keep us healthy. They may be the way that we respond most quickly physiologically, is the changes in the microbial world, and that might be a good thing. But we shouldn't discount that. I mean, people are always thinking, "Oh, this is really, this is going to be really bad," but I think we should keep an open mind to that they might be a great part of the solution.
Absolutely. I mean, there's a lot of evidence that microbes are involved in some of their metabolic byproducts in cloud cover. And that certainly affects climate.
Yeah.
You already mentioned the idea of the permafrost releasing methane, arguably a 50 times more potent greenhouse gas than CO2.
Yeah.
But I think that some people, and this takes us back to what we were talking about before this began, with this idea of people getting siloed, they'll say that, microbes are okay no matter what. I say, first evolved, last extinct. And that's true. But we have to live here too. Mm-hmm. And microbes probably could shift and adapt and change, but it wouldn't be the world that we want to live in.
Mm-hmm.
So understanding what aspects of the microbial world are associated with what makes our world good to live in would be a really good idea.
Yes, absolutely. I think overall we just have to think more holistically.
I mean, we're all connected, all of us. And it's not a surprise that we are communicating with other organisms. I'm communicating with you now. Microbes are communicating all the time. It's like a cacophony of communication, and we know so very little about it.
Mm-hmm. Mm-hmm.
And you cannot help but think it's important when you look around. I would argue that sometimes we get ourselves into, I'm going to call it intellectual narcissism, single colonies, growth in pure culture. It's one of my heresies, as I call them. That's not how bacteria, for example, normally live. But we put a lot of emphasis on it, and it served its function. But now it's time to look at interactions between organisms. We wouldn't know very much about animal biology if we looked at animals of the same species by themselves.
Yeah.
And it's no different.
Yeah, yeah, absolutely, absolutely. And it comes from thinking of microbes as foundational organisms, not ways to understand transcriptional regulation, which has been fantastic. Obviously, we couldn't have done it any other way. And that's true of organismal biology. Microbes started everything, and so they're foundational for microbiology as well as for understanding transcriptional regulation. Also, the other thing is that, it's either they're thought of as transcriptional or as pathogens. And, to this day, so there was just, can I say this? You can cut it out if I can't. But in Science Magazine, they had a little news and views sort of thing in which they were announcing something called the immunome. And what they're trying to do is they're apparently millions and millions of dollars are going out to describe the cell-cell interactions and the molecular interactions that go on across an immune system, right? And that was a 3-page long little news and views announcing this. And microbes were not mentioned once, not once. And we know that they tune the mammalian immune system. They tune the immune system. It's probably going to be true that they tune the immune system of all animals. And it's just breathtaking that microbes were not mentioned.
So Jonathan Eisen.
Yes.
And Jonathan Eisen has an award he gives every year for the Bad Omics Award. So I cannot wait to send him a link to that, which I'm sure he's already seen.
Oh, yeah.
So, everything becomes popular. We call it scientifically sexy, whatever it is you like to say, and then people jump on those bandwagons. But really, the important things are the questions you're asking and the tools you have to analyze them. And one of the things when I teach microbiology, I don't have enough time to teach the adaptive immune system. But when I teach the innate immune system, that's preset. It's completely a reflection of the microbial world. I'm quoting one of your papers. And I am. It's just a reflection of that. And the best example of that ever, and I have been accused once in one of my evaluations that I have TCT on the brain.
TCT, yep, you do. It runs the third wave of the mammalian sleep cycle.
So there you go.
Yeah.
So that story is important because it tells you that there was an expression long ago called PAMPs, pathogen-associated molecular patterns. And these are the things that let your body know that you've been infected. For example, lipopolysaccharide is a very potent pyrogen. It gives you a really quick fever at nanogram levels, I think. And it's part of the membrane, the outer wall of many bacteria. For example, if you take insulin, you'll notice it says no endotoxin, and that's what they're referring to. Because you need to know that. The innate immune system recognizes it like a spring-loaded trap, and we have a particular response. So it turns out that there's a particular kind, and this is a lot of work that you did, so I'm going to wave at you about it.
Yeah, one of my students coined the word MAPS, yeah.
Yeah, so...
Tanya Koropatnik.
So MAP stands for?
Microbe-associated molecular patterns.
And that's because it isn't just about pathogen. I'm staying away from the whole idea that many people have, that the word pathogen, hi Arturo Casadevall, many people don't like the word pathogen because it's all about context, right? For example, you have E. coli in your gut, which is necessary for the production of some vitamins. However, if you're in a serious accident and those same bacteria are released into your peritoneal cavity, you'll have sepsis. So we accept that. But it turns out that many of the same mechanisms that we're talking about for recognizing pathogens also were important for other kinds of associations.
Yeah, that's how they evolved for my money.
Yeah.
Because it's, I often, when I give talks to physicians, I'll say to them, so how many bacterial infections have you had in your life? And they'll, obviously you took antibiotics or whatever, but how many have you had? And they'll say, 20, 30, whatnot. Every single day of your life, from the time you were born, you are interacting with your normal microbiota. And the most parsimonious explanation for what drove evolution of the immune system is the interaction with the microbiome.
And it surprises me a little because we have no trouble saying that the temperature is involved with that. Oxygen levels are involved with our evolution. Nutrients.
Yeah.
Why not the microbes that are swimming around us in the air and the water all the time?
Oh, yeah. Well, and the adaptive immune system, for my money, as Mark knows, evolved to be able to take care of complex consortia. You had to have a memory of your normal microbiota. And so, think about it. Every day, the cells of the adaptive immune system turn over rather quickly. So they're always saying, are you okay? Are you sure you're okay? Do you know everybody? Okay, you know everybody. It's a situation that is really, and it had been thought that the adaptive immune system was a non-self-recognition system. Well, if you agree that your microbes are part of self, it's principally a self-recognition system, and that it also takes care of non-self. One of the things that's fallen badly, and I just actually wrote a paper about this for PLOS Biology, is that there's a shared derived character of all vertebrates is the carriage of a consortium. And coincident with that is the presence of an adaptive immune system. And it was thought that the invertebrates did not have any control over, and Charles Janeway, very famous and very august immunologist, had the mistaken notion that invertebrates didn't need an adaptive immune system because they lived for very short lives, had lots of young. They were what is called by ecology R-selected, live fast and die young. But there are invertebrates that live for 250 years, and they don't have a very complex microbiome. It turns out that invertebrates have a different strategy, and that is that instead of using a complex microbiome, they very often have a very simple microbiome. And not only that, it is stable. And so they don't need to keep remembering and remembering and remembering. So one of my favorite examples is the honeybee. And Nancy Moran has shown that the honeybee has 5 phylotypes. Every worker honeybee around the world has 5 phylotypes, individual to individual, reproducible. The little guy that I work on, Euprimnoscolopes, a little bobtail squid, has 1 species of, and it can be strain specific too, of Vibrio fischeri. And it has 1, and it collects it from the environment every generation anew, and it's that this particular species arose from other Euprimna species that were older that are thought to have started the symbiosis. And so it's probably been with Vibrio fischeri, this little guy, for 5 million years as part of the larger Hawaiian archipelago. 5 million years. So it has selected, it has about 4 generations a year. So 20 million times those generations have selected that microbe from the environment with fidelity against a background of a huge number. The Vibrio fischeri is relatively rare in the environment, and so they have to do a huge selection, which they do within 3 hours.
So it's interesting because we recently heard a little bit about the light organ of Euprimnoscolopes. And I always shake my head. The one time I went fishing for squid, did you hear about this? Mm-mm. Ned didn't make fun of me?
No.
So I caught 1 in a dip net. There it is, that big. And everyone's out there catching lots of stuff. And I go, Ned, I got 1. He goes, you're doing it. Keep going.
That's great.
Yeah, I'm not a good field biologist. But the point is that they don't develop that light organ until they see the bacterium. So the bacterium is giving them a signal.
Well, although they do develop during embryogenesis that really complex juvenile specific organ. And that juvenile specific organ has a morphology that's associated specifically with colonization, with getting those bacteria in. And you see that those 2,
yeah.
I'm pointing to There's a picture over there that shows these two really complex fields, ciliated fields. And what happens is there are 1 million bacteria per mil outside in the water that are not specific, and Vibrio fischeri is like 100 to 500 of them. And within 3 hours, that ciliated field on the surface of the newly hatched baby light organ collects Vibrio fischeri and resolves it outside the tissues as the only partner that can come in. And they make little aggregates of cells, and then they get a signal from the host, and that's the chemotext chitobios, and they dive into the light organ.
So there's this communication between both partners. That's the part that fascinates me. When I would teach about symbiosis and I would talk about, we know about integrated symbioses, like chloroplasts or mitochondria, that have been so fully integrated you can't cultivate them. And you might, and I've had students do this, look at the Squid-vibrio symbiosis as a, quote, simple version of this, because you can cultivate both sides of them and you can manipulate certainly the bacteria, and there's some efforts to manipulate the squid as well. But the issue is looking even at that like less associated, less integrated form of symbiosis. There's so much interplay of signaling.
Oh, it is breathtaking. In that 3 hours, when those little groups of bacteria are hanging on outside in that ciliated field waiting to go in, they change gene expression and adapt to a migration path they have to go through that is on fire with antimicrobials. They sit there and adapt to low levels of those chemicals in a mucus matrix. And they require a capsule that Karen Weissach is mentioned, or she has spent her whole career working on this capsule. It's wonderful. It's not a capsule, it's an exopolysaccharide. And so, but they then, are talking to one another, and they attach to the cilia of that field, and they talk to the host, and the host changes gene expression by interaction with 5 Vibrio fischeri cells on the surface on either side of the organ, on average 5 at 3 hours. 5 cells in a background of 1 million others that are coming in and out of their body cavity.
That is a whisper.
That's a whisper. It's a whisper. I am here.
Yeah.
Yeah. And boy, it changes. You get a robust change in gene expression. I had a great postdoc, Natasha Kramer, who worked with myself and worked with a guy over at the University of Kiel. And it was just a wonderful, wonderful collaboration with Philip Rosenstiel was his name. And it was just, we couldn't believe that 5 bacterial cells could trigger a robust change in gene expression across the 20,000 cells of the juvenile light organ. It's amazing.
So it's about listening. And that's, again, something that many people, and incorrectly, would say is a more casual kind of situation. And it makes you think about the organisms that live within us. And there's been a lot of work being able to show how some microbes in your gut are recognized as friend as opposed to foe. And some people on this campus have worked on that, where you can start to change one aspect or another of this and can evict things or allow them to stay.
Yeah. Oh, yeah. It's a very dynamic situation. And, it's really, really, really fun. The bacteria talk to their hosts. And we know from Jeffrey Gordon's work, for instance, during in mammals, during embryogenesis, the embryo develops receptors on their surfaces for their microbial symbionts. If they are not infected with their microbial symbionts, those receptors go away. And so in the studies that we do, when the bacteria then go into the crypt spaces, what happens is they, from the crypt space, deep inside the tissues, they signal the loss of those ciliated fields that are responsible for colonization, because they're all about getting the right bacterium in. You don't need them anymore because the microbes are with the host throughout its life.
So So many cool titles for this kind of work. So many great things that I've seen people being kind of silly about. You know, there's a horror film called Let the Right One In?
No.
Yes.
How cool.
So, I mean, that needs to be a modified movie poster for your lab, don't you imagine?
Yes.
Just like Tales from the Crypt, by the way.
Yeah.
Because that's what you want to call the region
That's
the
right.
light organ develops.
That's right.
So,
That's right.
and this is part of the joy of microbiology and macrobiology. See, Margaret's fortunate because she is not only, and I love this expression, and it will embarrass you, being called a thought leader.
Oh, you're very sweet.
But it's true. Some people have a lot of influence in the way, I mean, I can shout my head off. It doesn't have the impact. But see, you do. And this is important. You can hear how much Margaret cares about this topic.
Well, actually, you know, one of the really pivotal opportunities I had was I had a fellowship. And during that fellowship, I decided, you know, it was a year-long thing, and I went all over Europe and talked to people about what they thought about microbes and whatnot. And then I came back, and I decided to have a meeting as to pay the it was actually Guggenheim, and to say to the Guggenheim, there's fruit out of your investment. And I had a meeting of 25 scholars from around the world, and we actually put a paper into Science magazine, and it sat in Science for a very long time. And then Science magazine kept trying to say, you know, we want you to focus this down on development or something like that. And I said, no, there are 5 topics we're covering. We're covering the origins of the earth. We're covering genomics. We're covering developmental biology. We're covering physiology and ecology. And we're talking about, you know, how microbes are foundational. And we came out with this paper as a whole group. It was the only meeting I've ever been at where nobody talked about their stuff. They wrote a paper. Each of those 5 groups would go into a room, and they had to come out with 700 words and a figure in 3 days. Wow. Yeah. And so we did this, and we put together Animals in a Bacterial World, which is a paper that we put into Science, and Science just pulled us, you know, they pulled us right, left, and center. And at the end of the day, I just got permission from all the rest of the authors to pull it from Science, because it was just not happening. And Dave Carl at the University of Hawaii was very kind to shepherd it into PNAS for us.
And I will put a link to that paper in the show notes. It is so worth your time. Also, some folks at phys.org put together kind of an introductory Micronaut version of it that I think is really useful for your time.
Oh, cool.
Yeah.
Cool. Yeah.
You know, I'm watching the time on the wall, and we're almost out of time.
Oh, my gosh.
Did you have fun so far?
Oh, it's, you know, I feel extremely privileged. I feel privileged to be invited to do a podcast with Mark, and I feel privileged to be accepted by the microbiologist. A lot of animal biologists now think I'm a microbiologist.
Well,
you know, and it's just because, you know, the microbiologists have been very gracious to let me to hang out.
So for the most part, that's what I've always found about microbiology. And it's very accepting. You can find exceptions to that, of course. I mean, it's a human profession. But the number of times that I have to balance people who are being, and there's an expression my father always used, and it's appropriate. If he didn't like somebody, called them a jackwagon.
Mm-hmm. Yeah, that's great.
I have buttons that say no jackwagon. But for every one of those, there's a large number of, in fact, very, very wonderful, impressive people that are willing to listen. And I want to emphasize how important that is, because a lot of times we think that the ideas or questions we have are no big deal or they aren't important. So it's so important to be listened to. And that's one thing that I think integrating into, you know the word I'm trying to use. Those kinds of fields like symbiosis are good at that because they bring lots of different people together.
Yeah.
Good Lord, I haven't even talked about Hydra.
Well, one of the things that I would like to say to the microbiology community is you guys have to step up. You know, and what I mean by that is you have to become foundational. Mm-hmm. You have to reach out to the people who are doing animals and plants and so on. And I was talking to, you know, some of my friends in microbiology, and I said, you know, microbiology is foundational, and you keep saying that, Margaret. And they keep saying, yeah, you keep saying that, Margaret. Of course they are. We know that. And I said, yeah, but a lot of the rest of biology is not on board, and they're more and more and more biologists are coming on board, but it's really a big problem, and the microbiology community can't stay cloistered. You have to get out there and spread the word, because it's important.
Escape from the silo.
Yes, escape from your silo and stand tall, because microbes do rule.
Yeah, they do. And there's an expression, and I know that much of the time when I talk to you, you kind of think, oh, there he goes again, because I do.
I never do that.
But I have an expression, OMG, for overwhelming microbial greatness.
That's great.
And,
I hadn't heard that.
And another one is WTM. Instead of, forgive me, WTF, it's what the microbe? And this is one of the things when I teach microbiology, I'll bring up really interesting ideas. Like if you look at Deinococcus radiodurans, and you hit it with this huge amount of radiation that fragments its genome, and it gets put back together in like 5 hours. What, the microbe? And that's important to remember. And that also opens students up, because this is what we have to do. We have to hit them early.
Mm-hmm. Yeah. And that's why you need an introductory course that really starts with microbes as foundation.
I think so. And there are so many interesting questions that you could approach that way. But I'm preaching to the converted who actually converted me. So, Margaret, I want to say thank you very much for your time.
Thank you, Mark, for this opportunity. It was really fun. And it's always fun to talk with you.
And you can tell Ned that he can hide as long as he wants. I'm not going to track him down for this.
Oh, you're sweet.
And I want to thank you for your time.
Okay, you're welcome. It's great.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it, macrobes or not. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with insanely cool links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Margaret McFall-Ngai is at the California Institute of Technology in Pasadena, California. Many, many thanks, as always, to David Renata for superb editing, especially under challenging conditions like today, and Reber Clark for the wonderfully quirky music that brings a smile to my face every time. I hope that you've enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.